Two major types of T-cell recognized peptides bound to proteins of two different classes of MHC molecule. There are two types of MHC molecule called MHC class I and MHC class II. These differ in several subtle ways but share most of their major structural features. The most important of these is formed by the two outer extracellular domains of the molecule, which combine to create a long cleft in which a single peptide fragment is trapped during the synthesis and assembly of the MHC molecule inside the cell. The MHC molecule bearing its cargo of peptide is then transported to the cell surface, where it displays the peptide to T cells. The antigen receptors of the T lymphocytes are specialized to recognize a foreign antigenic peptide fragment bound to an MHC molecule. A T cell with a receptor specific for the complex formed between that particular foreign peptide and MHC molecule can then recognize and respond to the antigen-presenting cell. The most important differences between the two classes of MHC molecule lie not in their structure, but in the source of the peptides that they trap and carry to the cell surface. MHC class I molecules collect peptides derived from proteins synthesized in the cytosol and are thus able to display fragments of viral proteins on the cell surface. MHC class II molecules bind peptides derived from proteins in intracellular vesicles and thus display peptides derived from pathogens living in macrophage vesicles or internalized by phagocytic cells and B cells. Having reached the cell surface with their peptide cargo, the two classes of MHC molecule are recognized by different functional classes of T cell. MHC class I molecules bearing viral peptides are recognized by cytotoxic T cells, which then kill the infected cell. MHC class II molecules bearing peptides derived from pathogens taken up into vesicles are recognized by Th1 or Th2 cells. The antigen-specific activation of these effector T cells is aided by co-receptors that distinguish between the two classes of MHC molecule. Cytotoxic cells express the CD8 co-receptor that binds MHC class I molecules, whereas Th1 and Th2 cells express the CD4 co-receptor with specificity for MHC class II molecules. However, even before T cells have encountered the specific foreign antigen that activates them to differentiate into effector cells, they express the appropriate co-receptor to match their receptor specificity. The maturation of T cells into either CD8 or CD4 T cells reflects the testing of T cell receptor specificity that occurs during development and the selection of T cells that can receive survival signals from self-MHC molecules. Exactly how this selective process works and how it maximizes the usefulness of the T cell repertoire is a central question in immunology. On recognizing their targets, the three types of T cell are stimulated to release different sets of effector molecules. These can directly affect their target cells or help to recruit other effector cells. These effector molecules include many cytokines, which have a crucial role in the clonal expansion of lymphocytes as well as in innate immune responses and in effector actions of most immune cells. Thus, understanding the actions of cytokines is central to understanding the various behaviors of the immune system. And that's two major types of T cell recognized peptides bound to proteins of two different classes of MHC molecule. Have a great day.